Not all substances are moved passively across cell membranes. Many substances must be moved in a direction that is opposite to their concentration gradient. In other words, they are moved from a region of low concentration to a region of higher concentration. This process is called active transport. For this type of movement, cells require energy in the form of ATP. Most of the molecules that utilizes active transport are ions. Being charged molecules, it is difficult for them to pass through the membrane. They use ion pumps, which are protein channels that use the energy from ATP to transfer ions from one side of a membrane to the other against their electrochemical gradient. One example is the sodium-potassium pump. Sodium ions enter the ion pump. With the energy from ATP, it changes conformation or shape and releases the sodium ions outside the cell. Next, potassium ions enter the ion pump. The pump changes conformation and releases the potassium ions inside the cell. The sodium-potassium pump has the job of keeping the axon ready for the next signal or nerve impulse. Active transport is also accomplished by a different class of membrane proteins called carrier proteins or transporter proteins. A carrier protein binds to the substance to be transported and carries it to the other side of the cell membrane where it is released. There are three types of these proteins or transporters: uniporters, symporters, and antiporters. A uniporter carries one specific ion or molecule. A symporter carries two different ions or molecules, both in the same direction. An antiporter also carries two different ions or molecules, but in different directions. These three types of carrier proteins are also found in facilitated diffusion, but they do not require ATP to work in that process. Another type of active transport allows molecules and sometimes whole cells to be moved into the cell membrane in bulk. Bulk transport mechanisms involve enclosing of large particles or large quantities of smaller particles to be transported in their own small globes of membrane, which can then bud from or fuse with the membrane to move the substance across. The different modes of bulk transport are endocytosis and exocytosis. Endocytosis is a general term for the various types of active transport that move particles into a cell by enclosing them in a vesicle made out of plasma membrane. There are variations of endocytosis, but all follow the same basic process. First, the plasma membrane of the cell invaginates or folds inward, forming a pocket around the target particle or particles. The pocket then pinches off with the help of specialized proteins, leaving the particle trapped in a newly created vesicle or vacuole inside the cell.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior.
Endocytosis can be further subdivided into the following categories: Phagocytosis, Binocytosis, and receptor-mediated endocytosis. Phagocytosis or cell eating is a form of endocytosis in which large particles such as cells or cellular debris are transported into the cell. Phagocytosis is used by macrophages to engulf a pathogen.
The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle.
This video shows how an amoeba consumes two paramecia through phagocytosis. After being closed by pseudopods or arm-like projections of the amoeba, enzymes are released to digest the paramecia. Binocytosis or cell drinking is a form of endocytosis in which a cell takes in small amounts of extracellular fluid. Binocytosis occurs in many cell types and takes place continuously to get whatever nutrients and other molecules happen to be present in the surrounding fluid. Binocytosed material is held in small vesicles much smaller than the large fluid vacuole produced by phagocytosis.
Wants to take in enter by a process called pinocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pinches off.
Receptor-mediated endocytosis is a form of endocytosis in which receptor proteins on the cell surface are used to capture a specific target molecule. The receptors, which are transmembrane proteins, cluster in regions of the plasma membrane known as coated pits. This name comes from layer proteins called coat proteins, which are found on the cytoplasmic side of the pit. When the receptors bind to their specific target molecule, endocytosis is triggered and the receptors and their attached molecules are taken into the cell in a vesicle.
A specialized endocytosis called receptor-mediated endocytosis also occurs in cells. In this process, molecules of a particular substance, such as cholesterol, bind to receptors in the plasma membrane. The vesicle is formed as the membrane sinks in. Then a lysosome joins the vesicle and the digestive enzymes break down the substance, releasing it from the receptors. The substance diffuses into the cytoplasm, the lysosome reforms, and the vesicle, with now empty receptors, becomes part of the plasma membrane again. This allows for the cell to reuse or recycle the receptors.
Cells must take in certain molecules such as nutrients, but they also need to release other molecules such as signaling proteins and waste products to the outside environment. Exocytosis is a form of bulk transport in which materials are transported from the inside to the outside of the cell in membrane-bound vesicles that fuse with the plasma membrane. These vesicles are transported to the edge of the cell where they can fuse with the plasma membrane and release their contents into their extracellular space.
Of large molecules like proteins that cannot pass through a plasma membrane. The process of exocytosis enables the cell to pass its material out of the cell.
This video shows a paramecium expelling a waste. To summarize, cellular transport can either be passive or active transport. Passive transport, such as diffusion, facilitated diffusion, and osmosis, transport molecules from an area of high concentration to an area of low concentration. Thus, this process does not require energy. On the other hand, active transport such as ion pumps, transport proteins, and bulk transport moves molecules from an area of low concentration to an area of high concentration, which requires energy from ATP.